Dr. Shackman participated in UBRP. He was once in your shoes. He was a UBRP participant in the late 1990s. He completed his bachelor's here at the U of A with majors in MCV and biochemistry and chemistry in 2000, and he was named the outstanding senior in chemistry when he graduated. Dr. Shackman received his PhD then after that in analytical chemistry at the University of Michigan at Ann Arbor in 2005, and then he was a National Research Council postdoc fellow following his PhD. After that, then he became assistant professor at Temple University in 2007, and he remained there until 2010, when he decided to move into industry and he started his career at Bristol Myers Squibb. Dr. Shackman has won multiple awards at Bristol Myers Squibb, including those in leadership and mentoring and also in research and development. And he is a patent holder, and he has co-authored 33 manuscripts during his time both in academia and in industry. So please join me in welcoming Dr. Shackman.
Thanks for that very kind introduction. Just my tech check. You can hear me. You can see slides. All good? Perfect. So I have a fancy title here. It's moving molecules faster through pharmaceutical development, multidimensional chromatography. But it's just an overarching theme. We're going to have a race here to see who can do how many slides faster between the two presenters today. So should be fun. Let's see here. Go PowerPoint, go. All right. So I'm going to just rehash a little bit what you talked about. I'm going to go way further back. I'm going to go back to 1978. We're going to have a little brief fun history because as an alumni, I want to show you the career path I went through, which is very nonlinear. It's very multidimensional. So I was born here in Cincinnati, Ohio. At the time, it was very low tech, right? So the first bulletin board system, the first internet talking kind of happened. Pluto was named a planet for the first time, and the first baby was conceived by IVF. It was not me. I'm not the first baby of IVF. Technology-wise, this is when Space Invaders came out. So I actually have been going to arcades for a long time. Now they have giant screen Space Invaders. This is when it came out. I was only there for 3 months before, due to my parents' jobs, they actually moved up here to Michigan. We moved to Battle Creek, which is the home of Kellogg's. We used to be able to go through the Kellogg's factory and put your hands in the Frosted Flakes and eat them. They don't do that anymore, not just because of COVID, but because we realize sanitation is better. And around my second grade year, again, for job and career-wise, my parents decided, we're going to hop in this 1983 station wagon, my favorite Toyota wagon. We're going to drive all the way down with me and my brother. It was a painful 4-day trip, all the way down to Tucson, Arizona, my favorite home of all time. Just to give you, again, couched in technology, this is right when Windows was first released. So the very first Windows, 85. That's also when, unfortunately, the ozone home was actually discovered. But it was also the first use of DNA for a criminal case. So just kind of getting themes of the changes in technology that's going on. This isn't actually me. My parents didn't actually have a good picture of this, but this is actually what I actually looked like about in second grade when my parents, who were very tech forward, said, okay, we're going to get a computer in our house. Like, this is a big thing in 1985. They got us a leading edge. It was a PC clone. I don't remember the speed of the hertz on it, but 5.25 floppies. It had this monochrome monitor. And this is me being the happiest kid on earth that I had this computer to play with, even though it's all text. And I basically sat down on my own, just playing around, and had a book called GW Basic. And I just started teaching myself how to program back at the time. Most of my way, I was up in northern Tucson. I went to Donaldson Elementary, ended up at Canyon Del Oro High School, if any of you are locals, up on the north side. And when I graduated, I didn't know what the hell I wanted to do. So I went to U of A because it's local, and the Constitution said Arizona schools are nice and cheap, which was perfect for my socioeconomic level at the time. And so, not knowing what I wanted to be, I actually started off in journalism. I was going to be a photojournalist in 1996 when I joined U of A. The journalism department wasn't doing great. But my mentor and counselor at the time, Roxy Katz, many of you may know her, she did a fantastic job guiding me through and finding me the right major, which ended up being chemistry. Why chemistry? This is photojournalism back in 1996. You went into a dark room with chemicals. You played with light. She said, you're going to be an analytical chemist because you're basically going to do the exact same damn thing. I said, that's great. I can do that. She was also fantastic at making all my AP course credits cross-link and all my electives work out, which is why I have all the majors. I did do extra work. She just was a fantastic mentor and counselor. Technology-wise, this is when the first cloning happened. Dolly the Sheep, if you're familiar with her. Google search was invented, and the first genome was actually sequenced. For me, this is when I joined UBRP. My second year was actually 1997, my sophomore year. And I was a little bit different being a chemistry major. It's a little bit of a rarity in the UBRP program. But my faculty advisor, Bonner Denton, shown here, he had a lot of very good applied ways of using analytical chemistry in the biological sciences. And so we did a lot of different things. But his main thing was always saying, go faster, go faster, go faster, in his very heavy East Texan accent. And this is him sitting here with his rocket car, which you may be familiar with. He breaks world records out on the Utah Salt Flats on this, basically a little tiny car with a gigantic rocket engine. And as you can tell the way I talk, I learned from him, talk very fast, go faster, do faster analyses. And that's really been the heartbeat of everything I've done since then, is always trying to make things go faster in life from an analytical standpoint. So after I graduated at Arizona, Bonner told me, you should go to Florida. They have a really good analytical program. I know this great spectroscopist there. You can do some fantastic things. So I inherited the car. This is still the same 1983 Toyota Corolla station wagon that I hopped onto I-10, and I can actually say I've driven every single inch of I-10 now, going west for fun and playing in California, and as well as east for my graduate studies. Ended up at Gainesville, Florida. When I showed up there, though, the particular professor he advocated for, the projects just didn't sink to my heart. And what ended up happening was you interview lots of different professors and you find what sings to you, what is passionate. Don't just go because you're going to get a PhD. Do it because you love it. Do it because you want to make an impact, and that you're going to do this for a career, not just as a job. And the particular person I ended up finding was a younger assistant professor, who just converted from being an assistant professor, who was in the field of doing separations to study hormone secretion from living cells. What ended up happening was I ended up loving this and signed up with him versus the older, more established, well-funded guy. Technology-wise, this is also when Y2K was a bust, the first genome first draft, and the first podcast. Kind of interesting fun facts here. What did I start doing, though, was not just the instrumentation and the studies and the biologics side of things, but I continued to do the programming. That way back in, that little second grader, that was still in me. So a lot of my work was actually sitting in front of this Windows 95 gateway computer. It's a 46. I remember that one. And doing a lot of programming to do my data analysis, even though that wasn't the major thrust of my PhD, which was actually studying hormone secretion from islets of Langerhans. So 2 years after I joined the group, my boss said, "Oh, I got a better offer. We're all moving up to Michigan." I said, "Oh my God, I got to buy pants and a coat. Holy cow." It was a very stressful thing, but I came back to Michigan. You can see I've actually made a triangle now, started Michigan down here to the east-west coast. Yes, the same 83 station wagon made that journey all the way up north to Michigan. It went back to its home state, where, again, I continued to do microfluidics and studying of hormone secretion. So basically, our intent here, research-wise, and the majority of my PhD work was saying, can we isolate islets of Langerhans, which are these little microorgans in your pancreas that secretes insulin and glucagon is the majority. It's sort of your homeostasis regulating hormones. Could we actually isolate these things, put them on this basically a slide? Could we perturb them? Could we induce secretion? Could we get knockouts, all these other cool things, and throw them on a chip and actually monitor insulin secretion? And that's really what we did. And it was actually pretty cool because at the end of the day, we got to go on covers of journals and doing some really cool stuff. But behind it all, I was actually doing just a lot of programming, just having fun in the lab. So when I got done with that, I said, I still don't know what the hell I want to do. By chance and whim, a very wonderful lady came from NIST to present at Michigan, so always attend your seminars, and she was talking about the microfluidic program at the National Institute of Standards and Technology. I said, wow, that's fantastic. And I applied to get the Research Council postdoc, a 2-year stint there, and I actually got it. Unfortunately, towards the end of my graduate career, the poor 83 station wagon, the number of engine fires was going to be a little bit much for us, so we dumped the car. I ended up borrowing at this time, my now in-laws car. We drove down to basically outside of Washington, D.C., where I converted to a Prius, one of the nice little 2005 Priuses. For metrics, I don't have this on here. The 83 Corolla got 227,000 miles on it, so yay. The Prius, way down the way a couple years ago, died out. It hit $297,000. So this is my application for buying, I guess, Toyotas. I drive a Subaru now, if you care. Technology-wise and in current events, this is actually Hurricane Katrina, the first partial face transplant, and the first YouTube video. So things are really progressing very quickly in the technology phase. When I first went to grad school, I had a stupid Nokia telephone, and now I'm starting to get my iPhone. I think I'm up to a 12 now. I actually don't even know what I have anymore. Again, doing microfluidics. I do a lot of this analyses and studying of cells and stuff on chips, but really where my passion behind it was actually in the programming. So all of the analyses, the image capturing, all that stuff was still kind of doing a lot of this back end stuff behind the scenes that actually takes up a lot of the stuff that you don't publish on. But at the end of the day, when you look at a publication, the majority of the work really doesn't make it in. It's all that little experimental section that you're really playing around with. So hopefully, as long as you're passionate about that, it's still a great payoff. After my postdoc, I still didn't know what I wanted to do. it seemed like the normal course of events should be, well, you're academic you should go be a professor. So I applied around, did the whole circuit. It was a painful thing. If you ever want to talk offline about the joys of interviewing to be a professor, I would be glad to participate about that. But I was actually very grateful and honored to get a position at Temple University in Philadelphia, where I started my own PhD research academic career, hosting a group there. And we got into this and we started doing sort of separations on chips, also doing some other more bigger instrumental stuff. And the hook for us at the time was working with collaborators. So as a young professor, you try to find the best collaborators that already kind of have CAN stuff that you can latch onto in sidecar so you can advance your program, get your grants. And we actually work on these cool little things called planaria and just looking at secretion from them, isolating them, doing knockout studies with them. Very cool stuff. But I wasn't doing it, which for me, as a passionate hands-on scientist, kind of sucked. I was teaching my graduate students how to build stuff. But really, I was spending about 90-hour, 100-hour work weeks as a young assistant professor. This was at the time of the Great Recession, writing grants all day long. And in the end, that turned out to not be my passion. Could have been different if my advisors previously had taught me how to write grants, maybe, but I was kind of teaching myself. So unfortunately, this didn't pan out for me. Technology-wise, this is the first iPhone, but oddly enough, you forget about this. Android OS was actually released at the exact same time as the first iPhone. So it's kind of good competition there. And alas, Pluto is knocked down to being a dwarf planet. So we've come full circle on Pluto too. So after 3 years, you come up for an evaluation after 3 years as a professor, and they say, do we want to kick you out? Is usually the way it goes. And I kind of turned the tables on and said, I'm cutting it off. I'm making the decision. I'm going to move on. And I applied around a little bit and ended up getting a position where I'm currently at. So the longest stint now, 11 years, is at Bristol Squibb. Technology-wise, this is the time of the first synthetic living cell, the first Instagram. I've just learned how to become a grammer. I'm old enough where I'm on that cusp between I should be Facebook versus I should be gram versus I should be something else and retired. The first successful SpaceX flight, so we all watched the Dragon doing its miraculous journey recently. And alas, the Sony retired its original 79 cassette Walkman. And if you don't know what a cassette is, I'm sorry. Fantastic drug. I'm not trying to sell you on it. It was an amazing thing, having seen what I've seen over these years of cancer is a death sentence. Cancer is just going to kill you. You get the big C, you are done for. And joining BMS, having my finger on the pulse of cancer research at the time, a drug that's actually, I can't actually legally say that it's curing metastatic melanoma, but it is. Amazing drugs going on, differences with immunotherapy, just a whole new technology. I can go and pull off my shelf all the books I took as an undergraduate at U of A. And I honestly, a lot of it is outdated and just not useful anymore. And I'm wondering why I'm even held on to them. I should have sold them back to the bookstore because technology is advancing so quickly. So, be aware of what you're doing right now, but also be aware of the changes and what's on the forefront because it's really amazing stuff. So at BMS, again, what we try to do is to discover, develop, and deliver innovative medicines that help our patients prevail over serious diseases. BMS used to be into consumer products. I've actually got some mineralized sitting in a corner over here that's made by BMS. Over the years, they've actually divested a lot of it and then really focused on really 4 key areas of disease prevention, the biggest of which currently is cancer. But it takes a lot of time and effort and resources, which goes against my mentality of faster to do this. I don't want to get into too much of the politics of, drugs should only cost 30 cents versus the other. Let me just give you some metrics from my current viewpoint, which is the drug development is a huge attrition risk. It is a gamble. It takes a lot of resources to do it. We're trying to make this faster, but the estimated rough time, and obviously things go faster and slower, is about 10 years to develop a drug from thought process to actual on market, like actually on the shelves or in the hospital as a non-clinical drug. And the cost to develop this drug, again, wide ranges. The error bars on this are huge, but the average right now, roughly about 1.3 billion from first initial concept all the way out to the marketed product. And it takes a lot of time and people to do this. And you can see here, this is 79. Actually, the graph I couldn't find was 78. When I was almost born, or just shortly after I was born, 100 million to develop a drug all the way out to this 2015, 6 years ago already, to 1.3 billion. Just big money that goes behind this. And a lot of that's because the huge amount of attrition. You start with 10,000 compounds, that's your ideas. That quickly gets knocked down due to toxicity, usually, or efficacy to 250. You go to clinical trials, maybe the efficacy still isn't there. Maybe you have off-label effects, maybe drug-drug interaction, whatever it may be. It just gets winnowed down. So, you get down from 10,000 down to 1 relatively quickly, and all that money in R&D that went into those other $10,000, that's just kind of lost. So it takes a lot of time again. So preclinical, 3-4 years, proof of concept, your clinical trials, that's sort of 6 to 8 years. There's not a lot we can do other than, have a pandemic where you could actually bypass a lot of that timelines and have a lot of research behind that, all the way out to manufacturing filing. There's a lot of, a lot of regulatory stuff, which is good. I love the quality controls. I love the safeties that we have in place, especially here in America, as well as some of the more advanced countries, to do this because it protects us. And, not just as a company, it protects us as human beings from off-target and toxic effects. What I do in pharmaceutical development is basically take this discovery stage sort of thing, very small amounts or library compounds, and we take them out to basically clinical, out to launch. So we have to redesign everything. What they did in medicinal chemistry usually is not going to happen out there in the plant. That's because if you remember undergraduate, hopefully you've taken organic chemistry, it actually looks the same at BMS, right? It's a bunch of synthetic chemists sitting there at the hood, doing little reactions in round bottom flasks and beakers and doing column chromatography and drying and stuff. It's all glassware. It's not that different, actually. It's just a lot more of it. But what changes then is when you get out of these round bottom flasks, you're going into reactor scale things. You're making big amounts of things. And this is where the engineering side of things comes in it, because you can't just make a huge round bottom flask. It doesn't happen that way. You're doing a lot of engineering scale up things. And you're also doing a lot of analytical chemistry behind that to understand everything about everything that's going on. So that's my job. So we characterize all the starting materials, all the reagents, everything that's going to happen along the synthetic route, all those streams and things going in and out of tubes until you get to that final active pharmaceutical ingredient, the API, the drug substance. So what we do in my department is we're supporting that route process development and optimization. We're supporting the commercialization and ultimately the transfer. So things that I do in my little lab ultimately becomes characterization techniques out there in the real world on global sites, and across many nations. So this is my simplistic. I'm an alkyl chemist. I'm not a synthetic chemist. This is how I think of chemistry. A plus B goes to C. Yay. I can still do that in my head. That's hopefully high school chemistry. Unfortunately, my synthetic chemists, as they remake the discovery route, they typically fail at the start. They make maybe only a little bit of C, maybe a lot of D, something that they really didn't know they were supposed to be making. It's usually a long synthetic route. So you're starting way over here with common things. You're starting with, say, benzene or some small molecule, and you're ultimately making Taxol, some gigantic molecule. There's a lot of off-target impurities. There's a lot of byproducts. There's all sorts of stuff. And a lot of times they try to make C and nothing happens. They're sitting there at this yellow, oily mass going, what the hell did I do? Like an undergrad studying organic chemistry that's about to go get an F in lab. We've all been there, I know. So what we do as alkyl chemists is we have to figure out how they do that by doing all the characterization. We tell them everything about their input materials. We tell them everything about their output materials. What's in it? What's the purity? What else is in there? What is the quality of the reagents? What is the form? How much other water is in there? And ultimately, when we get to characterizing the actual API, we have to say, well, is there anything in there that's really, really, really bad? So talking about part per billion studies, trying to talk about, in the media right now, it's a lot of nitrosamines off sartans. So we have to find those part per billion trace methods to be able to say, yes, it's good, or no, don't send this out. And it's tough because we don't have infinite resources. That $1 billion is spread across the entire 10-year timeline, across hundreds of thousands of people, really, in terms of person hours. And so we have to come up with methods that are very quick, sort of quick decision-making methods, characterizations, all the way out to those methods that are going to go out into the real world, the ones that the FDA is actually going to look at, or other health authorities, and say, yes, you can use that to say this drug can go on the shelf. So it's really tough. And from my mind as an alkyl chemist, is the error bars start really big, and then they sort of crimp down until when we get to something that's going to go into an actual person, those error bars are as tiny as possible and as well characterized as possible. So we have lots of cool new tools to play with, things that I would have never imagined in my wildest dreams as an undergrad, taking instrumental analysis with Bonner. There's things like super high resolution mass specs that cost $500,000. There's brand new detection from very simplistic analytical techniques. GC is a very old established chemical analysis tool, but we're innovating, coming up with new ways of looking at the molecules coming off the back end of it. We do much more things in silico, so using our computers to model things. Other types of cool new ways of just looking at molecules by hitting them with different sorts of laser focus pulses to break them down and see what's going to happen with them. NMR, which is very common in organic chemistry, we're now looking at basically doing reactions and running it through the NMR in situ so that we can actually watch reactions at that time scale of the reaction. We're not just doing liquid chromatography, we're not doing gas chromatography, we're doing supercritical fluid chromatography now. And my heart and soul right now has been doing multidimensional chromatography. So marrying two different types of techniques together. So chromatography, if you're not familiar, this is my one slide to hopefully ground everybody back out. Its name means color writing, invented about 1900 by Mikhail Sweat, shown here in his fancy picture. And all he did at the time, even though this was groundbreaking, this changed the world of analytical chemistry at the time, and organic chemistry for what it's worth, was he basically took a big tube, just like he did in organic chemistry. He packed it with chalk, basically, and he ground up a bunch of plants, put it at the top, ran some organic solvents through it, and he saw, wow, I'm getting these different bands through it. What is going on in my big tube, shown here actually graphically, as I do this? And see if my fancy technology is going to work with me. There it goes. Oh, he puts his tube up there, and what he sees is these bands. What are those bands? They're different chemical components separating as they run down that tube because they're interacting with the tube differently because they're different chemical compounds. So we have what's called the stationary phase, and then the solution washing through it is the mobile phase. And so maybe chlorophyll B really likes to stick to that chalk, and this keratin doesn't really care about talking with the chalk. It just wants to run down really fast. So that's how you separate things. And a little fun bit of trivia, he was destined to do this because his name in Russian actually translates to roughly color. Today, our kids, like this really cool brownie, should have been Nobel Prize sort of stuff. Our kids are doing this all the time right now in their STEAM creative projects. Bring your kids to work day. We take, coffee filters or paper towels. We'll rub a magic marker on it. We'll stick it in a solvent and watch it do some really fantastic art. But that's effectively what FET was doing at the time too. Is you're actually separating things based upon the interaction with the solid versus the liquid phases. So pretty cool stuff. My kid loves making art projects like this. She just came back from camp yesterday with a nice tie-dye shirt. What is that? It's chromatography. We don't do it with big tubes anymore for analytical chemistry. We actually do high pressure or high performance liquid chromatography, nominally called HPLC. And what we've done is we've actually just shrunk down that big tube. We made the particulates inside of it much more homogeneous, much finer, so that they're much more efficient. What that means is those bands that are separating happen faster, and they also are much more narrow. So it's easier to discriminate between them much more easily. And a lot more, computers and stuff behind it to make everything all automated and detection along the way. So, you can imagine rather than my big tube, me looking at it with my eyes, we now have a computer that says, okay, once it gets through this tube, what do those bands look like once they come off the end? And they digitize that, and then we can quantitate those things. So that's one-dimensional chromatography. It's been around for 60, 70 years. More recently, in the past decade or two, we've really seen out of the translation of academics into the industry, the use of multidimensional chromatography. So once things come off that first column, we can then do something very different and translate that into a second column. So maybe one is packed with chalk, and maybe the other is packed with, a resin-based gel or something totally different. So you get very different separations on it because the molecules going through are going to interact with it differently. What's cool about that is once each band comes off the first dimension, you can actually cut that out and just look at that one band versus looking at the entirety of everything. So if you're familiar with 2D polyacrylamide gel electrophoresis, that's a very old two-dimensional technique where you're looking at a big square, sort of like this. What we do in 2D LCs is we don't just look at the entirety. We actually cut things out and separate them individually, one at a time, and then paste it all kind of back together. So here's an example of how we use this. We have this particular API, this active pharmaceutical ingredient. We want to characterize it. And what we find is when it breaks down, it creates this nasty degradant, something very toxic. And we say, well, my one-dimensional chromatography, it's not separated. There's not any selectivity there. Okay, and if I take this API and I actually formulate into the drug product, it's going to have other stuff in there, other active ingredients, other things in there, all these other components, so that when I degrade this thing, not only am I missing it, but it's getting to be a very complicated one-dimensional chromatogram. Okay, so what if I had a second dimension, and let's just look at that second dimension unto itself, and it can resolve or separate out the degradant in the API. Great. And if I just ran that second dimension method and I put everything into it, I'm still, it's very difficult to understand what's going on in this region. I wanted to see the degradant, but there's all this other junk in there. And why is that? Because all this other stuff is still in there. Okay, let's look at this differently then. What if I just took that first dimension chromatogram where that API and the degradant are stuck together, but then I transfer it to that second dimension, but just that individual peak. That's on them, just cutting it out, and I transfer it. It's all automated with a commercial system. And now I take that one-dimensional peak and I put it on the second dimension. Now I can actually resolve out the API degradant without all the clutter of all the other garbage that was in that sample. And it makes it very easy to quantitate and actually track this degradant throughout the life cycle of the API. So it basically removes the noise by only focusing on that one important peak that's coming through. We also do this in terms of things where we just can't figure out the separation because it's just impossible to do it in an easy to do way, so that we can do fast and actually get some data off it. So there's this kind of, I don't want to say new class, but an underrepresented class of impurities that happen in APIs called atropisomers. And what that is they're sterically hindered molecules. So you can imagine this molecule here, it wants to rotate around, but the little functional groups sticking off of it whack each other. And so it can ultimately rotate if you give enough energy, but it's very slow. And you can actually have these hindered bonds of rotation creating atropisomer isomers. So these impurities here, basically once it rotates, it creates a new form. It's the same structure, it's the same molecule effectively, but it's just rotated a little bit. But they have a lifetime that can be minutes, hours, or even years because of that steric hindrance. So they're actually physically resolvable and can physically interact with your body differently once you inject them or ingest them. So we actually have to track those things. It's really hard to separate these guys by an LC method. Very tough to do. In this particular instance, we had these four components that we try to resolve. Here's our main API. We tried to come up with a method. We got two of them resolved out of there, but one of them was just sticking there. We couldn't figure it out. We had another method that did a really good job on pulling that one atropisomer out. It wasn't so good on the other two. So, if we just put these two things back and back together and just put everything through, it would just become a mishmash. So how do we fix that? How do we fix that? We do it by two-dimensional chromatography. So we take this first dimension, but we just look at the big main peak. We excise out. We just want to ignore all these other guys that we've already separated. We know whether or not 2 and 3 are there. And we only look at the mixture of 1 and the API and atropisomer 2. And we transfer that down to the second dimension. And now we can actually track all 4 of them using that multidimensional chromatography. And so just kind of visually looking at it here, we actually do it by slices just to see how it's tracking through the main peak. And again, it's relatively quick, but it removes all the clutter. And that's why we love it. And it's also the same sensitivity as if we'd done that one-dimensional chromatography. And what's really cool for me as a scientist is all this stuff it's made by Agilent, which is a very large instrument company. They have all this turnkey. You can run the hardware, easy peasy, great. It'll do everything. It's sitting there on the screen. Data analysis, frankly, sucked. So we're sitting there in BMS going, how do we fix this? This particular graph is actually made in Excel, of all things, because it was terrible to try to use Agilent software. And we ended up just sort of back-ending and writing our own software. Where did that software come from? It came from my grad school project. It was the exact same software code, just basically taking that and tweaking it to a new commercial thing. So even though it's stuff that I'd written 10 odd years ago that I was still passionate about, I get paid now to actually still play in that domain, which makes me super happy. So this is me. This is actually the references from that atropisomer article that came out in, a couple months ago, 2021. This is me, the happy kid, referencing a grad school paper that I've still been programming on. I still do it on my own home computer. Every day I look for new challenges, new hackathons within the company, trying to still play with this. I'm still super happy doing my day-to-day job, but it's this other little add-on that makes this the sugar coating on the job that keeps me there and keeps me energized about it. So I've been all over the place in my life. I've done academic, I've done effectively government in this, and I've also done industry. I've seen the 3 things. So don't be afraid of the nonlinear path. You may not end up in the spot that you think you're going to be at. Maybe when I was in grad school, I said, I'm going to be a professor. I'm going to be Professor Johnny. Wow, that sounds kind of cool, right? That's not me. That's okay, because I'm super happy where I'm at now, and I'm still making a good difference in what I do every day. You want to explore diverse concepts, technologies outside your home field. Multidimensional chromatography is not a pharmaceutical technique. It actually came out of the petroleum industry. Multidimensional separations came out of biology. It was polychromatic electrophoresis that got it started, and that's why we're still here today, is doing because of those early experiments. And also find unique ways to use your strengths and passions. Maybe your day-to-day is you're pipetting all day, great, that's fine. But maybe you're back in at home is like, I really love machine learning. That's huge. If you can find a passion in that, you're going to go so much further than people that just are sticking within their own little blinders of this is what I should be as a biologist or a biochemist or even an alkyl chemist. So you're not going to recognize any of these names. I put this on there because they've done so much for me, so I want to make sure they are there. But I want to point out these 2 wonderful, wonderful ladies, Jessica and Caitlin. They were the interns that came to BMS. They are the ones that made this happen. They came up with a lot of the workflows that allow us to do this on a day-to-day thing. So if you are interested in the undergrad intern program at BMS, feel free to share my email, Jennifer. It's great. Currently, obviously, we're in summer now, so it's not an option. But next year, we start looking towards the end of the year. Hit me up. BMS.com also always has it pretty well as a clickable link to find the intern program there. Highly recommend it. when you're not doing UBRP, of course. UBRP is, of course, the best program ever. So thank you. That's my slides. And I think I hit my time limit too.
Right on. Thank you so much, Dr. Shackman.